So last up is Cecilia Aguero. And she's had the hardest job of anyone in our lab for a very long time, which is trying to figure out what the gene that's controlling Pierce's disease resistance is. Sounds so simple, doesn't it? She's been working, how many years now? 10 years? More than 10 years. 11 years working on this project. And it's very difficult because although it segregates, it varies as a single gene would. Is that little Sophia? Hi, Soph. How are you doing? She's too heavy to lift now, that's the problem. Where was I? Sophia cast my thoughts away. Yes, thank you. So although Pierce's disease seems relatively simple as a single gene that segregates that way and it appears when mapping to work that way, genetic mapping, if you actually go to the next level of figuring out what the sequence is and where it is and how it works, it's not so easy. So it's working as though it's a whole complex of genes and we don't know what it's complexing with. So she was tasked with that task. Unhappily, or maybe happily, who knows? So she's going to tell you a little bit about that and her time in the lab, too. Come on up.
Good afternoon. I'm going to be repeating all the things that have been said, and on top of that, showing exactly the same pictures. But you'll have to bear, it's our way to say thank you. And I am happy that independently we have arrived, all of us, to the same conclusions, right? So it must be true. So first of all, the title, The Wish to Sequence Andy's Genes, is a little bit pretentious, but it would be so nice, or when I was preparing this talk, I was thinking, what are the attributes that make Andy such a good breeder, such a career that he has built? So what are the attributes that make this man? And I will repeat all the things that you have said, maybe with my bad English. So the instructions were to say, what did we learn when we were working with Andy? And the first thing that I thought is, I was his student first. I took 101 B. I was a graduate student from Argentina, and it was a pleasure to go to his classes because it was a break from the other classes that were more formal. We were outdoors. We ride the van to the field. And I enjoyed a lot his expertise and his passion when he was teaching grape growing. The only problem was that he was speaking too fast, and still he speaks too fast for me. Can I change the... Oh, sorry, and what I regret is I didn't take the 101A class, because Andy is one of the best ampelographers in the world. And I found this funny quote in the internet. Andy could have identified a specific varietal with his eyes closed and upside down. So several years later, I joined the Walker Lab. It was, again, a pleasure, in this case, not to go to his class, but in this case, to go to work. And other people have said the same. The environment, the work environment, could not be better. And that speaks for your leadership. And all the people that were in our lab from different parts of the world, from South America to Europe, China, and you said, I think, 80 masters and PhD students. So that's incredible, and I think that everybody is so proud. Everybody that was in your lab is proud to be in your lab. And even my husband, when he introduced me, he works in the industry in Napa, he says, this is my wife, and she works for Andy Walker. Like, he's proud of that too. He steals my credentials. Yeah, you can see that the map there with all the pins from people that were coming from the lab from everywhere. And I think that as a researcher, he was in a very interesting time because to the classical breathing, all these new molecular tools were being developed to assist the grape breathing. And he succeeded Dr. Olmo, that is shown in those pictures, and another legend of grape breathing. And we share the same major professor, right? Carol Meredith. So a lot of our work was in the field, the classical breathing, the emasculation of the grapes, but then a lot of work in the greenhouse, a lot of work in our lab. And I remember the day that the sequencer arrived to our lab, everybody was so happy. But again, so much work, and I want to recognize the work of Dan, Nina Romero, and Ron Hu. Everybody have said that, but without their assistance, not a lot of things could have been accomplished. They were wonderful. And on my side, I think it was a similar journey. I started working with tissue culture, micropropagation of grapes, embryo rescue for table grape breeding. And then the next step was to go to transformation, to genetic transformation. And this was like 20 years ago, and we were so excited. And at that time, we were, and even a few years ago, we were transferring big pieces of DNA into the grape genome completely randomly. But now we have these new gene editing techniques that are revolutionizing the genetic work. And one of our last experiments was to knock out an aquaporin, that's a protein that's in the membrane of the cells and is involved in water uptake using the CRISPR-Cas9 system. And we are studying the plants that were obtained with the Macaron and Bartlett labs, and we are getting very interesting results from that. So very specific, guided and site-specific genetic modification. But going back to the breeder, to the work that the breeder does, is very complicated, it's very complex. Not only because he has to evaluate so many things, not just the resistance to fan leaf, but then the material has to root well, otherwise the nursery will not use it, graft well, etc., etc. And on top of that, all not the technical and scientific aspects, but the socioeconomical aspects of grape breeding. Grape is very complicated because at the end you need to sell the wine. So the grape breeder needs to have this vision. He can look 10 years, 20 years from now. It's not just a scientific problem. It's much more complex than that. And I think that Andy has been great at that, at connect to the industry, communicate with the industry. One more time. And who doesn't know Andy Walker? I was thinking of that title for my talk too. Because you say I work with Andy Walker and everybody knows who he is because of all this work that he has done for the industry and with the industry, right? And the future. Maybe I like too much science fiction, but I think that as we talk about precision viticulture, in just a few years we will be talking about precision breeding, and we will be able to use all the integrated data that is coming from the transcript, the analysis of the transcriptome, the proteome, the metabolome, etc., we will be able to integrate and be able to end the puzzles that we have in system biology, and somehow we will be able to target, to produce a targeted modification in specific type of the gene, the genome, right? Could be a gene or not. And so maybe we are able to have a Cabernet 2.0 if we can just go and touch the little parts that we need to modify. But that work, even this science fiction breeding, would not be possible if we don't have the diverse genetic material to work with. And I am very happy that Luis used the word treasure today, because all the material that was collected by Andy and all his collaborators is a treasure for UC Davis and for viticulture in general, and we need to curate that material. That's a real treasure. So I want to finish with a toast for your incredible and fruitful career. And I hope that now that you have a little bit more time extra, Viv and you will visit Mendoza. Thank you, Andy.